So today I'm going to share some of my experiences, and I'm here because I interacted with Kyle as well, and it was because of a meta-analysis. So that's another reason why we should know more about this approach and how to do sciences using meta-analysis. And because we published a paper in 2023, and Kyle, while doing another work, found our paper, I could interact with him and learn with him about meta-sciences and have more quality in my meta-analysis and being able to be here sharing with you some of our results. It's fantastic. So I really recommend you to dive into that and learn more if you can. And as we were introduced by Mariana, I define myself as a freshwater ecologist. So that's my background. So I work with plankton in freshwater ecology, and we try to understand the impacts of human presence in different aspects and how plankton respond and interact with these new pressures we are facing nowadays. And I'm learning with Kyle how it is to be a meta scientist. I need to tell you that I wasn't aware of this before meeting him, and I'm learning a lot. And maybe that's a new research line you, as researchers and scientists and students, want to pursue in the future. So there's also another way to do science nowadays that is not only in the lab with experiments or in the field, but also using data. And we have a lot of good quality data nowadays we can use to answer questions in ecology and freshwater ecology. I'll let him introduce himself later, but just to let you know that I'm also learning with this process. And it looks like it's a long way to go, but you're going to see that time is going to fly by and it's going to be very quick. We are just like touching the top of the iceberg, the tip of the iceberg. So we are not going to dive deep into the subjects and all the ideas and the procedures of doing a meta-analysis, but we're just going to have a general idea of what it is and how to start and how to be careful while doing it so we can generate a good quality product, a good quality paper or thesis or synthesis. So that's our aim here. So we are kind of give you a general idea of everything, and then you can learn more and um, learn with the papers. We are going to circulate a bunch of papers, and this presentation is going to be available to you later. So you're going to have ways um, to follow up and learn more with other materials. So we will have this general overview of meta-analysis. Then later we will have a hands-on part with Kyle, and we will have time for questions. But please feel free to write your questions, your doubts on the chat, so people will summarize them and ask us. Unfortunately, we don't have enough time to answer all the questions while it goes, but that's the purpose of being here, interacting with you. So if you feel comfortable writing the questions, please do it. Or you can also send us a message later and an email or reach us out through SIL, and we will be glad to talk to you and answer the questions as much as possible. So first of all, I bet when you were thinking about attending this webinar, you were searching around and trying to know more about meta-analysis. What is it for? How are people using it? Is there a reason why it's becoming so popular nowadays? So you probably have a general idea what it is. And what I'm going to bring today is what it is and what is not a meta-analysis, why it is important to integrate a meta-analysis in your current research agenda, and then give you some ideas on how to evaluate. What if you don't want to use a meta-analysis, but you want to read a meta-analysis and understand what is being cited or mentioned, or at least tell if it's a good or a not good, so good meta-analysis. So at least a critical thinking about what it is and what it's not a meta-analysis. So why people are doing it? Usually people are doing it to generate insights without needing very substantial resources. I can tell it myself because when I first got my position that I have now, it was 2018. And in Brazil, we were facing a very difficult political situation. So we didn't have money for research. We were like, we were employed without resources for research. So I had to move forward to build up my team, my students, with no resources at all, no grants at all. And that helped me a lot. That was the way I could move forward and start doing something and start my group without resources. So I can tell from my own experiences that it's a way if you are facing a lack of resources or if your grants are small enough so you can't go to the field or run some lab experiments, you can at least have, if you have a computer and internet and access to the papers, you can come up with a very nice idea and hypothesis testing, and you can have a very good outcome of these papers by doing a meta-analysis. Also, it involves low risk compared to fieldwork and experimental studies. So building up again from my own experiences, after I got the job, then we had to face the lockdown because of COVID-19, and I couldn't go to the lab. So I had the resources, but I couldn't go and reach the resources and run the experiments. So again, I was using meta-analysis to keep doing the research until I got everything open again, and my students could access the lab and go to the field and do other types of studies in the lab. So what if you can travel? What if you don't have resources to go to the field, or if you don't have the resources to buy the chemicals, or you don't have a lab or facilities to help you to run your experiments? You can decide, oh, I don't have enough resources, so I'm not going to do science. So what if you use meta-analysis to answer questions without having these resources? Again, it's another reason why. And also because we are living in a moment in history where we have a bunch of data. So information is not a problem nowadays, and access to information is less than before, but I think the quality of the information is a big issue. So we have tons of information, but we need to find ways to select the good ones and summarize what they are telling, or at least have an idea what have been done until now, and then we can have a talk and decide how to move forward based on what we already know. So at least we have an idea what we have, what we already know, and what we don't know. So meta-analysis can help us to answer some of these questions and have an idea on how to move forward from what we know. Also, people are using meta-analysis to support their ideas, their own projects, papers, theses, and sometimes they are citing that they don't have a meta-analysis in their field, and this is a good argument. So saying that we don't know enough, it's a good reason why you could have your project granted, you could have your paper accepted, or at least considered to be accepted, because you're bringing information or necessary information to fill the gaps. So having a meta-analysis is good, but not having one is also good. So you can tell that we don't have enough to tell about a certain area. So those are some of the reasons you might have found while you're searching around and trying to find meta-analysis in freshwater ecology. And I couldn't bring all the papers together. I was thinking about doing how can I say, a systematic review again and update the information, but time was flying by, and we are all also very busy with teaching and other stuff. And then I remember this paper from Sara Lodi, Godoy, Ortega, and Bini that was published in 2021, and they made a good overview of what has been done in freshwater ecology. And also another paper that came out this year by Pietro Polo and Nakagawa and a big group of meta-scientists telling about the quality of meta-analysis and what we should care about in meta-analysis in ecology and evolution. And why I'm mixing freshwater ecology with ecology and evolution. So based on Lodi's et al., we saw that freshwater ecology correlates well with ecology and evolution publication. And we see that both fields are using meta-analysis to support their ideas, to test their hypotheses. However, when the paper was published, they noticed that papers in ecology and evolution, they have better quality. They report better the results of a meta-analysis than freshwater ecology. So it's a red flag for us So we want to help our group, to help our research area to improve. We should pay attention to how to report the data and make data more available to people following our steps. So we are doing meta-analysis, but we still have a way to go and to make better outcomes compared to our sister ecology and evolution research area. But we are on the way. And how we have cited this type of paper? Mostly, if we consider different article types, so we have empirical, meta-analytical, methodological, review, and theoretical types of paper. The first one on top here. I don't know if you can see my arrow. And the colors, the different shades of gray, they tell us where in the paper, the primary paper, a meta-analysis has been cited. So most of them use meta-analysis to support their ideas in the introduction. Some of them, but much less, use them in the methods because they are performing a meta-analysis or using an outcome from a meta-analysis. And some of them use the results in a discussion to compare their results or to have an idea on how other people have found similar results or to support their findings. But when we look at the quality of the data we are using to compare in the bottom, so the letter B here, we see that we are using it, but we are using less than we should. For example, we are missing the gold part. We are missing the quantitative results. We are missing the outcomes, and we are comparing our results to a meta-analysis, but qualitative, not quantitative. So we need to use the outcomes to have a size effect to compare with the data we are generating by our own study. So we are on the way, we are using meta-analysis, but we need to extract more information from a meta-analysis to compare our results in freshwater ecology. And now comes the definition, and we'll start to talk about the definitions. And I see and I tell my students two main purposes of using a meta-analysis, and sometimes we misuse the definition of meta-analysis. You probably are going to find lots of paper doing systematic review and calling it a meta-analysis, or just a scientometrics and calling it a meta-analysis, or just an overview of the field and where they give their perceptions of the field, or sometimes a very senior researcher telling his experience about the field with some of data he gathered from the most known research groups, but without doing a meta-analysis. So in the past, it was very common, and I think people are getting more information and being aware of what it is and what is not a meta-analysis now, and they are using it in a correct way. So it's just another red flag. Make sure you are really doing a meta-analysis and not some sort of review or qualitative review of the field without doing the proper approach. So when we talk about meta-analysis, we usually have the statistics on our side. So we extract from the primary studies some what we call effect size. So quantitative information in a way that we can later combine the results from independent studies, and then we can standardize this information from the individual studies to a common unit. And we standardize it in a way that we have not only the outcome, but a certain measure of error, like standard deviation, standard error, confidence interval, and most importantly, the sampling effort. So we have to standardize them in order to compare and calculate a summary effect, like a combined effect of all the papers that we found in our search. But they have to be in the same unit, and they have different weights. Because if a study samples more, it has a better sampling effort. It would count more than a study that just sampled once or twice. So we need to find ways to give different weights to the precision of the study and the sampling effort it has. So we use statistics to help us to organize our findings and standardize our results, and then calculate a summary effect or a general effect or a mean effect. And I'm going to show you later on a figure and what it is. So it's not a very new approach. It was built on the knowledge that was gathered in medical sciences, psychology, epidemiology back in the 1970s. So it's quite like well-known approach. But now it's becoming more popular because we needed to summarize and synthesize the knowledge we already have. And it's often a follow-up. So it's the next step after we do a systematic review. It's not necessarily made based on a systematic review, but most of the time when we want to synthesize information from the literature, we need to have a protocol to search for the information and how we decide if that information is useful or not. And then later we are able to put everything together, and then we can kind of have a summary of everything and a synthesis of everything we got together. In the literature, it's funny that they use some of the examples like mixing apples and oranges. So you need to be very careful not to combine studies with different messages, outcomes, information that later will be very important for the outcome we are generating. If you don't have this criteria of combining different sorts of studies or different primary studies, we won't have a very good synthesis. So it won't be representative of the question we want to answer. So we need to be very aware of what kind of what criteria we're going to use to include or to exclude the studies that are going to be part of our summary. And some people say that it's many articles in one. It means that in a way that if you got together all the studies with the main message, the similar messages with different outcomes, and you just want to have a general idea what we have so far, then we can have a synthesis. And then we can tell, oh, what we know so far brought us to the point where we can decide if we are going to use this approach for the future, or we have enough information or not to move forward from where we are right now. So it's kind of a summary of the literature, and we can build after this main outcome, this main summary from a meta-analysis. You probably saw something similar to this figure, and in meta-analysis we call it a forest plot. So in a forest plot, we usually have the combination of the independent studies that we call primary studies. So after searching and having our search criteria and inclusion and exclusion criteria, we came up with a number of papers we consider would be similar to be combined in a synthesis. And then we kind of extract the outcomes, the quantitative information we want to extract from them. And then together with this outcome, we extract information about the errors, standard deviation, confidence interval, any sort of measurement that states the error associated to the measurement, and the sampling effort. And with this information, we can then later give weights, different weights to the different studies. For example, here we have 5 studies, and the size of these boxes here, they represent the sampling effort. So if one study has a larger sampling effort, the size of the box is going to be larger. So this study in particular is going to contribute more in our summary effect, which is the combination of all the primary studies or a summary of all primary studies. Then, for example, the study 5 that has a very low sampling effort and has an error associated to it. So here, when we have all the results combined, standardized in the same unit, we then can calculate what we call general effect or summary effect based on the individual effect size. And we have different ways of combining these results by taking into account the variability inside of each one of the primary studies and between all of them. I'll give you an example. The other day I was teaching this to my students, and one of them comes from medical school. And he was telling me how it is, for example, to run the cohort and the trials. If they are trying to find a dose, for example, for a medication or a new treatment, they need to be very rigorous and very strict to a protocol. So they kind of run that protocol several times with a very similar group of people. So they tested several times in a way that's very rigid. They have to follow all the steps. So they are really aware that the protocol is very restrict, and the studies, they don't have variation between them. So the only variability they are going to find is going to be to the people that are involved in that trial. So for example, if we have 5 trials, we know we are using a very standard protocol and following all the rules properly. So all the variation we have is not because we are using a different method, but because people will respond differently to the dose or the medication, and that's what we want to know. So in this case, when we know that we have low variability between the studies, we can calculate what we call a common effect, and we use a common effect model. So all the variability that we find is due to the population itself, or the variability within each one of the studies that we got with our search. However, we know that in environmental sciences and ecology and freshwater ecology, this is not something realistic because we are working in different parts of the world. We have different ecosystems. We have seasonality. For example, how can I compare lakes in Brazil with lakes in Canada? And they have a very different dynamics. They have different and complex interactions. So we have some similarities, but also lots of differences. And sometimes we know those differences exist and we want to measure the difference between them. For example, if we want to compare carbon emission from lakes in temperate, boreal, tropical ecosystems, we know that where they are in the world, and because it's a proxy for the seasonality and all the other things that could interfere in the results, we want to measure how they are going to respond and how much is going to be the gas fluxes or the gas budget, the carbon budget from those ecosystems, considering they are in different parts of the world. So what we do is we know there is a variability within each study and between the studies. Then we run a specific type of model taking this into account, the variability within and between. Then we run a random effect model. And later, after calculating the summary effect, we can analyze the variability between the studies and see if there's a pattern. Could it be an answer or related to the seasonality or to the latitude, for example? So we can try to explain the variability with some other information we have from the studies. And later on, you're going to see that we call it moderator. So we have some sort of variabilities that we are aware of, and we want to see how much they explain of the variability we have when we run and we perform a meta-analysis. So that's how we combine the studies. We have a synthesis for the studies, but we also can explain the variability we are finding between the studies. So it's not only a synthesis, but a way to take the information from the synthesis and then move forward and try to explain the variability. So 2 main purposes, important purposes for us to use meta-analysis as a resource. And we should tell people this. We should tell and measure this variability by using, for example, I squared, tau squared. There are some ways of making it available to other people in our papers. So we need to make sure we are stating this, making it clear and transparent about our measurements so people can build up based on the results we already have. And here about some, just one example on how we use this, and we compare the variability and try to explain it in a paper that was performed a couple of years ago by Fernanda Cruz, and I helped her with the meta-analysis, and Dr. Philippe Junot from Montreal. And she was doing her PhD, and she was dealing with pesticides removal by aquatic plants. And she wanted to know, because everybody was telling her, oh, we know that aquatic plants, we call them also macrophytes, They can help to remove pesticides from the water. Oh, everybody's seeing this, lots of results showing the same trend that they can remove it. But we didn't have an idea of how much was it. So we decided to combine the data in a way that we could have like an estimate of how much this removal was and if it was significant or not. And later on, we were trying to explain the patterns that we found, the trends that we found with the variability between the studies. So what we found was that considering all the studies that met our criteria, we came up with this number that is 38, like almost 39% of removal. So when we have experiments where we have controls without plants and treatments with plants, those with plants, they're removing more pesticides from the water than the controls. And this removal, considering all the studies and the outcomes they brought, again, weighted by their errors, associated errors, and sampling effort, was around 38, 39%. So at least we had this estimate, and now we know how we can talk to people and tell how much is the effect of using macrophytes or aquatic plants to remove pesticides from the water. And because she had other information from the studies, we could correlate this and try to explain the variability between the studies with the moderators. So we understood the contribution of the pesticide type, the species that were involved in the experiments, the importance of the concentration that was used in the treatments, and we could explore other information just based on the outcomes from the papers we got in our meta-analysis. So it is just an example, a simple example on how you can use it. You can use this, for example, to justify a grant. For example, what if we didn't have a decision here? What if our summary effect wasn't conclusive? So it was equal zero, we couldn't tell anything that it was positive or negative. That would be a good reason why we should invest more into experiments in measuring the effect of plants on pesticides removal, for example. And then we have other purposes as well to use meta-analysis. So we can determine if we have a controversial findings. For example, a long time ago, we wasn't sure how much and if there was a contribution from freshwater ecosystems to carbon emissions. So we didn't know how much carbon was emitted and how much carbon was buried in the sediments. So we didn't have the budget. And now we know, and we are contributing to IPCC and other important reports that deals with climate change. So we have at least an idea. It's not perfect, but at least we have an idea. And we also have an idea of what are the factors affecting this carbon emission or burial in the budget. So we can tell based on previous studies. We can have an estimate, for example, of this. Is it higher or lower compared to other ecosystems in the world? For example, do the ecosystems in Brazil, they store more carbon than those in temperate lakes or not? And if yes, what is the size of the magnitude of this storage? Is it too much? And why it is too much in comparison to other ecosystems? So we have a way to compare our data. We also have ways to compare patterns, for example, differences in effects. As I told you, we can use the moderators to explain the variability. We also can replicate our data. What if you are implementing a new method in your lab and you are not sure if the method is going to work? So you have to run several pilots beforehand, before going to the field and make sure that the method is going to work in the field. So you have independent pilots and you want to combine it in order to make a decision if you're going to use that method or not. Then you can use the meta-analysis. And this is the case where you don't need to have previous studies published in the literature to combine them to have a meta-analysis. So in this case, you are combining results from your lab and you just want to make a decision based on what you already have. So it's an useful approach as well. And also, if we have enough data about some subject or a topic, we just want to make a summary of it, so we don't have to go to the primary studies every time. Of course, we need to go and know the primary ones because they are sometimes they bring more information than just the meta-analysis. But if we just want to have a general idea, sometimes we can use that kind of synthesis, and from there we can move forward and have other questions regarding the results we already have. And this is useful, for example, if you're using a meta-analysis to write a perspective paper, or if you want to give advice on how to improve your subject or how to improve your research area, and what do we need to get better if you are not having good results, how can we improve, and how can we advance some sort of areas in our field. So it's very useful in many other ways than just synthesizing an effect or answering a question. It goes beyond that. And now we are gonna go to another part of the presentation where I'll make sure that you understand that meta-analysis is not a systematic review, but we sometimes need a systematic review to generate a meta-analysis. So sometimes people just want to make a qualitative synthesis, and they want to make sure they are getting all the information in a way that people can follow the steps. We are, in this case, using the scientific method. So we are searching papers in the literature, and we are making sure that anybody that wants to follow our steps or do it again, they are going to find similar results. So it's just a protocol for searching and including the data we want, and then selecting the information we want, but not necessarily doing it, following this protocol brings you the outcome necessary to run a meta-analysis. We understood that a meta-analysis deals with outcomes, quantitative outcomes, and we have to weight the outcomes by their sampling effort and errors associated. So we use a systematic review to get this information. But if we stop here, let's suppose you decide you wanted to perform a meta-analysis, but then you find out you don't have enough results to move forward to the statistics. For example, you don't have enough examples, observations, degrees of freedom to run a statistical test. And then you can stop here. You can use just a systematic review and tell, I wanted to move forward and perform a meta-analysis, but we don't have enough data to do that. So this is what we have so far. And you can tell people what we need to have a meta-analysis in the future. So you can use the systematic review to state the art and to tell people what you already have and how. And we need to work in order to have enough results for the next step if we want to answer a question that has this that wants to answer this kind of question. So here is just like it is very simple. This is one of the most simple ones I use with my students. We have better versions, fancy versions, but what is most important is to tell people and be transparent on how many papers you got from the bases, Web of Sciences, Scopus, Google Scholar. Let's say, for example, in Brazil, we have the SIL that is now part of Web of Sciences. So you can search in PubMed, and you can tell people you went there, you search with a very specific search screen or search code that Kyle is going to work with you in the afternoon. And based on the papers that we retrieve from the search, we can apply our inclusion and exclusion criteria. And those that meet our criteria, we use them in this following step. So we can remove the repeated observations, repeated papers, we call them duplicates. So we remove them, and we start screening in a way that we go first to the title, the abstract, the keywords, and see if it's reasoning, it makes sense what that paper is talking about. It has the information we need, and we select this. And later on, with the ones we select at this stage, we come back and read the full paper and start extracting the information. And we can extract the information we are going to use in the next step, that is a meta-analysis. But sometimes we have the paper talking about what we need, we have the information, but we can't use the information because it doesn't bring the full information. For example, the sampling effort, we don't have an error measurement, so we can still exclude papers in the end. So usually you start with thousands of papers and come up with a synthesis that has like dozens or at least like 50 papers, and it's very disappointing. And some of my students, they say, oh, come on, we had 7,000 and we just used like 50 papers to write the summary. And these 50 papers, you might think that, oh, it's not enough, but think about all the criteria they need to meet to be there. And we want to make a good summary with comparable results. So we need to make sure that 50 ones we are using, they are good quality papers. So that's why we are using this protocol, to make sure we are not combining apples with oranges and that we are using good quality information. And then based on this, making good decisions in our work. And another thing that people sometimes mix up with meta-analysis is scientometrics. And I learned with Caio that they call it in meta-science scoping reviews or systematic mapping. So people, based on the information they get with the search protocol, for example, Prisma, they can have information about some specific research area and how people are dealing with results from that area, who are the main groups working with the subject, where they are located in the world, what sort of methods they are using, and what kind of subjects they are dealing with, and what are the main shortfalls. So they kind of use information about the studies to tell how to move forward or how to advance some area or research area or subject, for example. It doesn't necessarily use the information generated by each of the studies, but for example, it could use only the information about citation, impact factor, citation indexes, where are the journals where most of papers are published nowadays, this kind of information. And not necessarily you need to go and read the full paper and get all the information you need. You're going to see with Caio, I guess, but if not, you can search on the internet that Web of Science and Scopus. In the end of the search, you can have an option called Analyze the Results. And if you click on it, you have a general overview of the keywords, the main journals, and how many papers per area. So at least you have an idea of if your search string and if the keywords you are using to search the preliminary papers are good enough, and then you can rethink your search. And but this is just an overview. You need to work better the results in order to have a scientometrics. And often, in order to perform a scientometrics study, you use bibliometrics. There are some sort of indicators that you can analyze the results and compare the results and tell about the topic, research area, and think about the outcomes we already have. So you can think about, for example, the contribution on the top, the collaborators, how the collaborators are linked to the other. Do we have like strong research groups, or it's very spread all over the world? People are connecting more broadly, or more, there are some, just like a few group of people working with that subject, this kind of stuff. And you usually use, as I said, publication type, journals, year of publication, research groups. And we have our packages dedicated to that, so you can easily have this information at hand when you decide if you want to do a meta-analysis, for example, or not. So it could be the first step for you to think about a subject or something, or topic you would like to dive into. And summarizing this section then. So usually a meta-analysis is a quantitative synthesis. A systematic review is a literature synthesis. a systematic review can help us to have a better meta-analysis and a better scientometrics. And in comparison to a meta-analysis and a systematic review, both scientometrics and bibliometrics, they are assessments. So one is a quantitative assessment, and the other one is more like broadly like a research that uses the indexes to tell us more about the research area. So often we first do a systematic review, and with the results we get, then we can explore more into a meta-analysis, a scientometrics, or a bibliometrics. But we can also do a meta-analysis without a systematic review and a systematic review without a meta-analysis. So those are the cases where we want to synthesize information from a research group, from our lab, for example, different pilots, outcomes, or in medical approaches, like where we want to find different outcomes from different populations, and we want to synthesize things that were performed with a very rich protocol that we know the variability is small. So we just want to do and calculate the summary effect. In a systematic review, we can answer broader questions as well without going to a quantitative synthesis. And in both cases, systematic review and a meta-analysis, we follow the scientific method. So we have a question we want to answer, we have a hypothesis, and we can test that hypothesis. And as we do in our labs or in the field, we have to find ways and design a protocol to search for the results, analyze the results, and answer our question. And if we don't answer our question with the design we made, we can change our question, or we can start again and try again and have more observations in order to answer our question. So we need to make sure we are following these steps so other people can do the same thing and reproduce our outcomes or get a similar result. And last but not least, I would like to mention that one of the collaborations I had with Kyle, and I'm learning with him a lot, is that how to report the results of a meta-analysis, how to make our data useful in a way that people can get not only qualitative but quantitative information from our meta-analysis. So it would be nice if we follow these steps, like a checklist on what we should report and how we should report, so we can improve our own papers and also be able to be critical about the results we are reading. Because before this presentation, I was trying to find examples in the literature, and I found a lot of bad papers. Like they didn't follow the protocols, they didn't report the results, and it's very frustrating. So if you want to move forward, and now it's the moment to discuss this, because, for example, if you are planning your thesis project, or if you are planning a research grant you want to submit to a research foundation in the future, or if you are thinking about your next project, next manuscript, think about the ways you could improve the data available to other people. And this is something I would like to share with you, because in the beginning, people thought that doing meta-analysis was not fair, because we were answering questions based on the hard work people had in the field or in the lab. But doing a meta-analysis is also hard work. It takes like years sometimes to summarize all the data or to extract the data from the preliminary study. So it's a lot of work, and it's necessary. We have lots of information, and we need to separate what is good information and what's good science from what is not, or at least we cannot use the information the way it's published. So make your papers more transparent, make it available, publish your databases in a way that other people can ask other questions with the same results you found with your studies. And thinking about in Brazil, where I work and how things are difficult for us in Brazil, it's very important that you value the hard work you had with the public investment. So you are bringing back to the society, to the community, the work you've performed and the results you had so other people can benefit from that. And it's a public investment. So everybody's going to grow with the information you got from your own paper and make it available to other people to ask other questions and advance the field. So this paper just came out. I think it's on the Environmental International website, but you can download the pre-proof, and it has a lot of like recommendations on how to publish your results and how to interpret the results that the published papers already have, and learn the good practices for your meta-analysis and systematic review. So these are our contacts, so please feel free to message us anytime. I have two emails because sometimes the one from the university doesn't work properly when I'm traveling. So please feel free to message me on both of them and Kyle. And thanks for having me. Thanks for listening to me. I hope I could send a good message to you. And I was sick a couple of days ago and I wasn't able to say things properly. I hope you got everything, at least the main point of everything. Thank you a lot.